Today we will talk about scientific research and medical advances. Introduce the latest scientific research on longevity and health, along with advancements in medical technologies such as gene editing, tissue regeneration, and stem cell therapy. 1. Human organoids. Human organoids are miniature, three-dimensional tissue cultures derived from stem cells. These tissues grow in a manner that can mimic the complex structure and functionality of human organs. Organoids can represent a variety of organs, including the brain, intestine, kidney, liver, lung, pancreas, and more. Their size can vary, but they are generally a few millimeters in diameter. Key points about human organoids. Origins. Organoids are usually derived from pluripotent stem cells, like embryonic stem cells or induced pluripotent stem cells, or adult stem cells. Growth and development. Organoids are grown in a special environment that mimics the conditions inside the human body. This involves a cocktail of growth factors and other molecules to guide their development. Over time, these cells differentiate and organize into structures that closely resemble the organ from which they are derived. Applications. Disease modeling. Organoids can be used to model human diseases, making it easier for scientists to study disease mechanisms and progression in a controlled setting. Drug testing. Before testing new drugs in humans, researchers can use organoids to screen for efficacy and toxicity. Regenerative medicine. In the future, it's hoped that organoids can be used for therapeutic purposes, potentially replacing damaged tissue in patients. Personalized medicine. Organoids can be derived from an individual's cells, allowing for treatments tailored to a specific patient's genetics and disease profile. Advantages. Human-specific. Unlike animal models, organoids provide a platform that is specifically human, allowing for a more accurate representation of human biology and disease. Reduction in animal testing. As organoid technology becomes more widespread, it has the potential to reduce the number of animals used in medical research. Complexity. Organoids can recapitulate the complex interactions between different cell types in human organs, something that 2D cell cultures can achieve. Limitations. Simplicity. Despite their complexity compared to 2D cultures, organoids are still simplified versions of human organs and might not capture all the intricacies of the real thing. 2. Advances in Alzheimer's research. Advances in Alzheimer's research. Alzheimer's disease AD, a progressive neurodegenerative disorder, remains one of the most challenging conditions to tackle in the realm of neuroscience. However, recent years have witnessed remarkable advancements in understanding and potentially treating the disease. Biomarkers and early detection. Previously, a definitive Alzheimer's diagnosis was possible only through postmortem brain analysis. Recent innovations, such as PET scans targeting tau tangles and amyloid plaques, are now providing the ability to identify Alzheimer's in living patients. Simultaneously, the discovery of specific cerebrospinal fluid biomarkers can predict the disease's onset even before symptoms manifest. Amyloid hypothesis challenges. Traditionally, the accumulation of beta amyloid plaques in the brain was considered the primary cause of AD. While amyloid targeting treatments showed promise in laboratory settings, many failed in clinical trials. This led researchers to diversify their approach, exploring other potential culprits, such as tau proteins, inflammation, and the brain's vascular system. Tau protein research. Tau tangles, another type of protein aggregate, have emerged as a significant focus. Some recent treatments aim to halt tau accumulation, with the hope that this may be a more direct route to slowing or stopping the disease's progression. 3. Brain-computer interfaces. Brain-computer interfaces, BCIs. Brain-computer interfaces, BCIs, sometimes referred to as brain-machine interfaces, BMIs, represent a direct communication pathway between the brain and external devices. This cutting-edge technology is rapidly progressing, merging the realms of neuroscience and engineering. Here's an overview. Principle and mechanism. BCIs work by detecting changes in brain activity and converting these signals into commands for a machine or computer. The brain's neural activity, often detected using electrodes, is processed and translated into tangible actions. Invasive versus non-invasive. Invasive BCIs, these involve implanting electrodes directly into the brain tissue. While they can provide more accurate and nuanced readings, there are concerns about long-term safety and potential side effects. Non-invasive BCIs, techniques like electroencephalography, EEG, capture brain signals without surgery. Though they're safer and more accessible, they might not offer the same precision as invasive methods. 4. Liquid biopsy. Liquid biopsy. Liquid biopsy is an innovative diagnostic method that involves analyzing samples of bodily fluids, typically blood, to detect and provide information about diseases, most notably cancer. Here's an overview of this groundbreaking technology. Concept: unlike traditional biopsies, which involve extracting tissue from the tumor or organ of interest, liquid biopsies examine circulating tumor cells, CTCs, circulating tumor DNA, XNA, and other biomolecules found in blood or other body fluids. Benefits: minimally invasive. Without the need for surgical procedures, liquid biopsies are less invasive, reducing patient discomfort and risks. Dynamic monitoring. The method allows for frequent testing, facilitating real-time monitoring of disease progression, treatment responses, and potential relapses. Tumor heterogeneity. Cancerous tumors can vary in their molecular makeup. Liquid biopsies can provide a more holistic view of this diversity, which can be crucial for targeted therapies. 5. 3D printing of organs. 3D printing of organs. 3D printing technology has extended its reach beyond creating simple objects and prototypes to revolutionizing medicine through the concept of bioprinting, specifically, the 3D printing of human organs. This advancement holds the potential to address the critical shortage of organ donors and transform the field of transplantation. Principle: bioprinting involves layer-by-layer deposition of biomaterials, such as hydrogels or bioinks, to create complex three-dimensional structures that mimic human tissues and organs. Personalized approach. One of the significant advantages of 3D printed organs is the ability to customize them based on a patient's unique anatomy, which could significantly reduce the risk of rejection and other complications. What do you think of our video? Let me know in the comments section below. Before you go, please hit the like button and subscribe to my channel. Thanks for watching.